Okay, so this is that lactose permease catalyzing the syn port of a proton and lactose across the membrane. Did I finish this part? Or did I just get to the page? Okay. Okay. Thank you very much. Totally blanked out. Okay, so this right here is going to have this carboxylate group right here. Remember, carboxylate is the carboxylic acid conjugate base, right? And this is a weak acid normally, right? Meaning that it's not a bad base. It's actually a pretty decent base. It's just usually acidic. So this H plus is going to attach from the outside. This is going to prep this for lactose to bind to it. And so lactose is then going to bind to it. Now remember, this H plus, that is your proton gradient that's going to drive this process, okay? Without that, you don't have the energy to do this process. This is why normally what would be a conjugate base for an acid is getting protonated, okay? It's literally because you have too much H plus, it's pushing that equilibrium in the opposite direction, right? That's the energy it's driving. Does that make sense? Okay. Now, this lactose is going to attach. You're going to get eversion. That is the correct terminology, by the way, in case you're wondering, what the fuck, did they forget the R? Because we don't actually go back to the same thing. Okay? So eversion rather than reversion, meaning going back to the same thing. Okay? Eversion, right here, this is going to mean you open it up into the inside. What's going to happen next is, you may have noticed, this was that lactose right there. That's not going to now be able to detach and go off into the inside. This right here is the protonated carboxylic acid. We're exposed to the inside now. We don't have a lactose blocking it. And so right now your concentration gradient is favoring release of the proton, right? And so we're going to release the proton. That actually is going to be spontaneous and provide energy, okay? And that was what was providing the energy for this. Is literally an equilibrium pushing this, right? Just an equilibrium happening from the outside to the inside, okay? Okay, and then this is going to release that proton. Now we have this carboxylic acid. There is nothing energetically favoring this. It's actually going to energetically favor going to the outside now, and this is going to cause it to evert or eversion of this. Does anybody have any questions of this process? Notice at this point we have quote-unquote reverted to the beginning, but it's not the exact same condition, so it's actually eversion would be the correct terminology. I do have a subconscious thing where I tend to say revert and invert instead of revert. It's a Freudian slip, right? Okay, so your specific channels, these can rapidly transport ions across membranes. We have ion channels. These are going to be passive transport systems that allow for the rapid movement of ions across membranes. Think a little bit like tunnels. The tunnels don't take energy to move stuff through them, right? They just facilitate the cars to move, okay? And so your ions are like your cars. They're going to be what provides the energy, usually by some sort of ion gradient, right? Sometimes set up by active transport, but sometimes not, okay? Now, the rates are going to approach the rates that we would expect for your ions diffusing freely through aqueous solution, though they will never actually reach it, because you do have an additional step, and that is going to cause a little bit of a delay. But with enough ion channels, that delay is very small. Okay? Just like with enough tunnels, there's no real traffic delay beyond the normal delay of roads, right? Okay. So action potentials are going to be mediated by transient charges and sodium-potassium permeability. The interior of a neuron contains a high concentration of sodium and a low concentration of a high concentration of potassium and a low concentration of sodium. Man, sometimes I get ahead of myself. Generated by the sodium-potassium ATPase. So you set yourself up to have a concentration gradient with an active transportation, right? The resulting membrane potential is going to typically be on the order of negative 60 millivolts, okay? Remember, this is one way we can measure the potential energy of something. Specifically because this is an ion potential energy, millivolts is really useful. because ions are highly correlated to electricity. Okay? Which is why you get this like incorrect statements of like electrical signals being propagated in nerves and stuff, because it has electrical potentials, but you don't actually have electrons moving through a wire or something like that, right? Electrons are not arcing through your nerves. You would die from that. Do you know how much electricity it takes to kill a human? 40 millivolts of electricity crossing your heart will kill a human. Okay? This is 60 millivolts. Every nerve has enough potential energy to kill you if it was actually expressed as current. Okay? It is not expressed as current. It's just silly. Okay? It'd be like every time you think, you'd kill yourself 1,000 times. Just stupid, right? Anyways, hopefully that has broken that thought process from you. Okay? This is a collapsing potential energy. You can think of it as like polarizations moving along. Okay? Like waves of potential energy. Nerve impulses, this is going to be your action potential. So this is going to be a electrical signal produced by the flow of ions across the plasma membrane of a neuron. Again, electrical signal is kind of the wrong terminology to use here. It's an electrical potential signal. Are you okay with me not making that clarification and correction? Okay. When you hear electrical signal, what are you actually going to think now? Electrical field signal, electrical potential signal, not any actual electrical signal. Okay? It's because biochemists rarely have much physics, right? And much like physical chemistry stuff. Okay. So the electrical signal produced by the flow of ions across a plasma membrane of a neuron, that is again your nerve impulse is one way to call it, but you can also call it an action potential, literally meaning that you have a potential that is being acted upon, right? The action potential is a rapid electrical signal across a plasma membrane. The action potential is generated when the membrane potential is depolarized beyond a critical threshold value, e.g. about negative 40 millivolts. Notice it's not the whole potential that was there, which was negative 60, okay? You can never get the whole thing. It would actually be really rough to reestablish that. But because remember, whenever you look at these things, percentages work in a kind of almost logarithmic manner, right? They have an exponential tails. So your membrane is going to reach approximately 30 millivolt before repolarizing. The amplified depolarization is going to be propagated along the nerve terminal. And by the way, the terminology actually gets better when you talk about specifics. So as they start getting into specifics in your book, the terminology clarifies. I just don't want you to do the overall picture and then forget. Okay? So it is a polarization, right? Just perfect terminology. Exactly correct. The membrane potential here in millivolts. And again, this is going to be your depolarization process right here. And again, see how it went from negative 60 up to the about positive 30. Okay? And then this is going to be your repolarization. And again, that's going to be pump, 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 and reestablish that polarization. Okay? And then this will be your resting potential right here. Notice that you're actually going to exceed that resting potential, and it will slowly drift back, and then you'll go ahead and reestablish it later on. Everybody see that okay? Okay, did I go off too far? If we went out further here, you'd pump more, reestablish this dip, and it'd go back. And you keep having to fight that, right? Because what would happen if we just let it sit? We'd go to zero, right? Potential energy is inherently unstable. Yeah. So if we just let it sit and there was nothing else blocking it, it'd go back to zero. So the transient charges in your membrane permeability to sodium and potassium ions, the membrane depolarization beyond a threshold is going to increase the membrane permeability to sodium. These sodium ions will flow into the cell because of the large electrochemical gradient across the membrane. So it further depolarizes the membrane, increase the sodium permeability. The membrane will spontaneously become less permeable to sodium and more permeable to potassium. The potassium ions are going to flow out of the cell. They'll return the membrane potential to a negative value again. And so this is that process we just described. Now we've described it in the exact steps. Okay? Okay, and so now again we're just going to see the individual parts of this, okay? Now we have an overarching flow to it. So this is the patch clamp technique. This is a method that uses a glass pipette to create a high resistance seal. We call this a giga seal because it's a really big seal for a cell, right? Between the pipette and a small patch of cell membrane. This actually enables the measurement of ion conductance here. And so this right here would be your low resistance seal. This is what a normal cell would experience, right? And so we take the pipette, we put it onto the cell. And what's going to happen is we're going to then apply some suction onto that cell. This is going to attach to the cell via that suction, right? Okay, and this is going to give us our giga ohm seal. Now, giga ohm, by the way, is an actual, ohm is resistance, right? Giga ohm being giga, the prefix, giga, gigabyte. So giga ohm. Okay, so we have our suction here. And so this would be the whole cell mode. And what's going to happen is we're actually going to break it open. It's going to spill the contents into our pipette. The glass will act as an insulator. And so now we can measure the electrical conductance of those ions, right? Yeah. And this right here would be the detachment by pulling. And so we don't provide any additional suction to this. We just pull, okay? And this is going to give us an excised patched mode, i.e. we're going to force this to become inside out, right? This is actually what you would see during this process. So remember that there's going to be some conductance equipment back out here in the part you're not seeing of that patch pipette. Okay? Typically you would have conductance equipment on both parts, by the way, because they're cheap. Unless you need to measure really, really sensitive stuff, which is this is not that sensitive. So the patch clamp method is going to enable the observation of your ion movements across single channels. Right here we actually can see closed versus open here. And this right here is the measurement of it. It's 4. Anybody know what the small p means? No? pH, POH, negative log. Yeah. PE, pH, POH, negative log of, right? Anybody know what an A is, capital A? Amps. Yeah, typically amps, though it can sometimes be activity, so do be careful. Okay. So this right here is, again, a 4PA. Again, that's a logarithmic scale, so 4 is actually pretty damn big, right? This is not amps, by the way. This would be your activities. Okay? By the way, 10 to anything changed in amps would be huge. Okay? Like, yeah, that'd be a lot. This is 4 microseconds, or milliseconds here, excuse me. This one would be the 400 milliseconds that you'd see. Okay, so this is again going to be your structure of your potassium ion channel. This is going to be an archetype for many different ion channel structures, and so you can use this to get a generalized idea of what the different ion channel structures would all look like. Okay? But this one specifically would be your potassium. Your sodium channel was purified based on the affinity for the neurotoxin tetrodotoxin from your pufferfish, actually. A little interesting side note, this tetrodotoxin, by the way, from the pufferfish. This is actually used a lot in murders and assassinations. When they use it in places like the Rocky Mountains, which don't have any pufferfish nearby, because when you look for something, you have to specifically look for it, right? You don't just look at the blood and then all of a sudden magically you get everything. That'd be ridiculous, right? Imagine all the data you'd get. They actually had a string of murders, like a dozen of them or more, that were all these tetrodotoxin pufferfish murders. Yeah. It was probably an assassination. Series of assassinations. Yeah. Anyways, it can be very difficult to detect it where you're not expecting it. So the sodium channel is going to be a single chain consisting of 4 internal repeats of similar sequences. Each repeat will contain 2 hydrophobic segments. These are the S5 and S6. These are going to form your actual pore. The 3 hydrophobic segments, which are S1, S2, and S3, these are going to contain the apparatus that will open the pore. 1 positively charged segment, that's going to be your S4, and this is going to act as your voltage sensors for your channel and will contain the apparatus that will open the pore. And so this right here again, so your calcium 2+ and your sodium plus channels actually share the same sequence pattern, and we can actually see these here. The potassium channel actually consists of 4 subunits, each homologous to one of the repeated units of these sodium channels. Your A mutation version of your potassium channel, this is encoded by the shaker, facilitated by its purification. So this right here is your sodium and calcium channels right here. Again, here's the S1, S2, S3, S4, S5, S6. Right here is actually the pore. Okay? And so we can see here, this is your 1, 2, 3, 4. What would those be? So 4 subunits, right? Each of them with their own S1, S2, S3, S4 portions or moieties, right? Okay. This right here is actually the shaker potassium channel. Yes, these colorings actually do match up, but notice that they're not exactly always, it's different because it's a different length, right? This is your bacterial potassium channel, and yes, again, these colors are similar portions of this. Okay? Okay, so this is the bacterial potassium ion channel right here. It's only going to contain 2 membrane-spanning regions that correspond to the segments S5 and S6. You can actually look down here. S5 blue, S6 yellow, and those are there, right? And then we have the pore segment. Okay? Okay. And so this is what this actually looks like. This would be looking down the pore, and you can actually see the pore, right? Okay? And then you look from the side view here, and this is what it would look like from the side. Okay? Notice that this part right here that's on top is this right here. The yellows are down here. See that? And they're in the depth of it. Okay. This is actually your single subunit of this. Right? How many of these subunits assemble? 4. You got it. That's your 4 subunits. Very nice, right? And you can actually kind of see how this would work, can't you? Okay, so right here, again, your structure of your potassium ion channel. This is again going to actually reveal the specificity, because when you're talking about a protein or enzyme, the structure tells you what it does, doesn't it? Okay, so what would you expect the binding site to look like for these things? What size would they be? Something that would match up with potassium. Would you expect them to match up with copper? No. Yeah. So the pore starts with a diameter of about 10 angstroms. It then constricts to a smaller cavity that's about 8 angstroms. The potassium must shed their water and interact directly with the protein to fully get through this. And this is the length, by the way, right here. So again, this is your selectivity filter, and that's that stretch of amino acids that's going to determine your ion preference. And again, that's that contracted portion that thins down. Okay? At that part, if you had anything that's significantly larger or smaller in atomic radius for that ion, it's not going to be able to fit through, right? Okay, and again, that's that stretch of amino acids that'll determine the ion preference. In this case, that stretch is TVGYG. And that's actually true for basically all potassium channels. They all tend to have that exact same stretch, okay? And the reason for that is this right here. Okay, so when we get in here, what's going to happen as we go from the large to the small, we're going to go, and potassium is going to have to lose its hydrated waters, right? It's then going to connect into ion dipole interactions with these groups right here. Anybody know what those groups are? The red. They're the oxygens. Black is carbon, blue is nitrogen, right? Okay. Notice tier, again, this is the tier right here, side chain. Everybody see it okay? Okay. These are those carbonyl oxygens in the mid linkages. Okay. Okay. These carbonyl oxygens are going to make that tetrahedral shape where it's that cage. Okay? And it's going to allow you to have the electrons, lone pairs, overlap with these potassiums, which is going to take over where the waters were, right? This is going to give you your complex with the potassium that's energetically favorable. Okay? Okay. Anybody have any questions about that? And again, what size has to match up here? Has to be the potassium without its waters, right? Okay. Now we will get to some places where it's like the potassium or calcium or magnesium with its hydrations. That would fit. We'll also get to some places where, notice like a iron 3+ is not going to fit well here, right? Okay. So different ion size, different ionic radiuses are going to be too big to fit or too small to be well bound to this. And so they won't lose their waters. They won't be able to fit through. So the large ions are going to be too big to enter your channel. So for example, we look at potassium here, and the ionic radius is 1.33. If we were to look at cesium plus, for example, is that going to be able to fit? No, it's just too big, right? So it literally can't get through. It just bounces off, even if it managed to lose its waters. So the small ions actually could fit, couldn't they? If they lose their waters, right? Okay. So how are they going to get excluded? Well, they can't interact with the selectivity filter because you have to overcome that free energy cost of desolvation. This is your hydration free energy, meaning it's the amount of energy it takes to strip those waters off that would normally be making that hydrated cage around the ion. Remember that hydrated cage, okay? Everybody understands what I'm talking about here? Okay. So what happens is, if you get too small of an ion in here, is it going to be able to make these interactions with these oxygens well? No. And so what ends up happening is maybe it can make one or two of these interactions, but you still have the waters and it's not going to be able to fit through, right? Okay. And so you'll try to get the, remove one water and attach, and I can't make the other interaction. The water stays on, I can't get through, it detaches, and then keep going, right? Does that make sense so far? Fantastic. Okay, this is actually what I was talking about, the hydrations. That's what an ion normally looks like in water, right? And so you'd have the normal hydrogen bonds past that, letting it solvate into water, okay? And this is what an ion looks like anytime you look at it in water. This is also why it typically raises the acidity in water, by the way. Okay, so this is potassium. What's the desolvation energy? Well, it's just the energy required to remove all of these waters, right? Okay. And the resolvation in the potassium channel is going to be these oxygens on these carbonyls doing the ion dipole interactions instead. Okay? And what do you notice about these energies? They're not equal. What's bigger than what? And so that means this process right here is spontaneous. Okay? So this process is spontaneous. You're spontaneously binding the potassium into the channel. Okay? By the way, the energy differences here, it's very important to get the right mixture of energy differences. What would happen if you had a huge resolvation energy here? Can anybody think what would happen? What happens if I take a bottle lid and I tighten it so damn much I can never get it off again? Am I ever getting that back out? No. So that would be what would happen if you made this huge. You would never get this back out. Okay? You'd have to use ATP or something to get back out, and you'd have to have a whole mechanism to it. Okay? This right here is sodium, by the way, and so sodium could fit through the channel, right? It's small enough, we'd be okay. But the desolvation energy is not overwhelmed by the resolvation energy. In fact, the desolvation energy is quite a bit bigger than the resolvation energy because it's not going to fit quite right into here. You won't be able to interact with all the carbonyls as you should. Okay? And what ends up happening is that because of this, you still have those waters attached, and it's not going to be able to actually go through. Okay? Now, the other way of looking at this is that the desolvation energy here, it's actually energetically favorable to go the opposite direction, right? And so what ends up happening is this tends to detach and go back in to the solvation, right? The other thing to think about here is, what if this was just a little bit short? What do you think would happen? So you may not get it flowing freely, right? Where it's barely any difference. But if you were just a little bit short, you'd actually start seeing potassium show up on the other side. Because, part of your population in any given solution is going to be high energy, right? Yeah. And so some of them would pop through. Okay? You need a pretty decent energy gap here for them not to come through at all, or almost at all. Mm-hmm. Yes, this one would actually be, assuming there's no huge difference in entropy and anything else, this one would be spontaneous. Okay. Yeah. But it may be some feedback on entropy. Okay, and some of these channels are actually driven by entropy. Okay, so this is your amino acids defining the selectivity filter. These are going to differ between the calcium and sodium channels because you have a slightly different selectivity, don't you? All right, and so this is your calcium channel, and you can see here there are 4 glues, which is kind of nice. Right here, this is your sodium channel, and you can see that you have actually a mixture. And so you have a D, A, or A, D, E key, K. So 4 potassium binding sites are going to be present in your selectivity filter. When a potassium ion moves into the selectivity filter, its positive charge is going to repel the potassium ion in the nearest site. This is going to push the potassium ions through your channel. You may have noticed when we were looking up here, how many are there here? 2. Yeah. So what ends up happening is essentially you get these bumping each other through. Okay? And so, again, the potassiums are positive. They're going to repel each other, right? And so this one up here, this is large enough that stuff is just going to keep bouncing in here and trying to push. Okay? And every time it comes in here, again, we have an energy difference that will feed energy into attaching, right? And so it spontaneously wants to attach. That's going to provide energy to hit the next one through. Okay? And so they keep going bang, 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 bang. Bang, 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 bang. Is that making sense? And basically every collision that has enough energy is going to hit this one through, and then it'll start spontaneously attaching. Okay. And again, that's the sodium channel. Okay, so this is your model here for the potassium channel ion transport. It's going to rely on the multi-ion binding sites within the pore. And so here you have your cell exterior, here's your cell interior. So understand that when we're doing these things, we're talking about an overall arcing concept, but we always talk about it in a specific so that we can actually have specifics to work with, right? That way we have a real experimentally determined channel that we've actually seen what it does, right? Instead of, I think it looks like this. Now, what does that mean? Well, in this specific channel, it's interior, exterior here, right? But is that actually meaningful when we're talking about the abstract? No, we could flip this, okay? It could be flipped, right? So I do need we need to concentrate on what we're talking about here, but when we're done talking about that, I need you to also have this like mindset, like I'm going to strip some of the specifics off and I'm going to leave it with an abstract as well, okay? Okay, now this right here is again the cell interior. We're going to have our potassiums. Why are there red rods on those? They're hydrated, right? That's the waters. Okay, so they're going to be banging around, right? And they have energy, okay? And so they're going to spontaneously diffuse, banging around, and one of them is going to hit this at the right energy, right? And what's going to end up happening is it will spontaneously lose its waters and attach to the channel, okay? Everybody got that all right? Okay, fantastic. And again, what is this channel here actually called? The specific name for the channel would be the pore portion, right? This is the pore right here, okay? Okay, i.e. the channel through the membrane. Okay. So the potassium is going to attach. Again, this is that binding site within the pore, right? And notice it's lost all of its hydrated waters, okay? They've rejoined this wet stuff out here. Okay. Now, what's going to happen is these are going to continue moving around, getting hit by the waters moving, all that other stuff, right? And one's going to come in with some extra energy, and it's going to hit, okay? And what's going to happen is this is then going to hit this, providing it some energy. This will then move along, okay? Now, please do note that because these are all basically the same binding sites, there's actually very small steps in energy to these, right? Right? And so what ends up happening is the energy difference between this, this, and this, and this, they're essentially negligible, right? Now, the energy for this one over here, where you have more potassiums hitting it, that one's higher, isn't it? So what you end up with is essentially an effect where this actually has a little bit more energy to it than these do, assuming that there's a higher concentration of calcium out here, okay? And then that's actually going to cause these calciums to sort of move down every time that another potassium approaches, okay? Now, it's not doing it because it says, hey, potassium, I'm going to move. It's doing it because of the repulsion, right? Okay. And so this is going to leave that site free. Once that site has been freed, it can actually start spontaneously doing the binding to the potassiums, and they'll lose their hydrated ions, okay? Now again, remember that once you bind this, you have a positive next to a positive, and what's going to happen? Repulsion, right? So that will move that down. We now have a free spot here. Well, that's pretty energetically favorable to move because you got these potassiums hammering on it. Okay? And you'll just keep getting that. Now, it does mean you typically form gaps in this inside of this pore. Okay? What would happen if our binding was too great? Right here. So the difference in that binding energy that we looked at right here, the distillation versus the resolvation within the site, this difference right here, if this greatly exceeds the repulsion energy here, this won't move down. It will never move off. Okay? That difference actually has to match somewhat with this repulsion difference. Okay? If it's less than the repulsion difference, it'll come off easy. And it'll pop right off. If it's just a little bit greater, you would need a pretty good amount of potassium outside of here to push it through. Okay? And that's actually what would determine how fast this moves through and what sort of concentration you need outside for it to go through. Remember that depolarization energies, now it doesn't go below this term and it doesn't go above this term? Guess what? That's what it's related to. Okay? That's the major driving force behind that. Anyways. Okay, so there's voltage gating. This is going to require substantial conformational changes in your specific ion channel domains. Your voltage gated channels are going to be channels that change conformation to a highly conducting form in response to changes in voltage across your membrane. Please understand again, I'm not going to make the full thing, but conducting means something a little different than normal conduction, okay? Again, we're talking about polarizations, okay? Some of the examples are going to be the potassium and sodium channels. The paddles, this is going to be domains that extend from the core of the channel and change conformations to open or close the channel. Literally, has anybody ever played pinball? Okay. You've all played pinball at some point? Okay. What does that kind of remind you of? A pinball channel? I.e., flip it upside down, you stand here, right? Okay? And then you use the paddles to block stuff coming through, right? Ding, 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 ding. Okay. Guess what? Somebody played pinball and named this thing, right? And it actually works, doesn't it? That makes a lot of sense. So paddles, like the paddle in a pinball machine, right? Okay. These are going to be the domains that extend from your core of your channel. They're going to change conformations to open or close the channel. They're going to be formed by segments S1 through S4. These are going to be, which are positioned to line in the membrane. So this right here is your S4 region. Is going to lie, there's your S4, right there. It's that red one. Everybody see it okay? There's, there. Okay. So again, that S4 region lies on the outer surface of your voltage-gated potassium channel. That S4 region, it's the voltage sensor. It's going to form an alpha helix that's going to be lined with positively charged residues. This is what it looks like from the side. Everybody see that? Okay. Note that it's actually oriented in the correct direction to match up with these figures. Okay? Which is beautiful. God, I love this book. The attention to detail is unreal. I could not write this book. I would screw this up, for sure. I'm going to be honest with that. So this right here, this is your S3, by the way, S2. This is your S6. Remember your S6 right there? That's the yellow part. There's pore. The white part being your pore. This right, or the gray, I should say. This right here is that blue S5, and those correspond to the segments we looked at in the one-dimensional drawing. Okay, so the whole thing matches all the way through that entire deal, color-wise, everything. Right. The movement of your S4 region is going to control, again, that's your voltage gate right here, your S4s. Okay, what are the charges on the voltage gate? Positive, right? Okay. So your movement of your S4 region is going to control the opening of the voltage-gated potassium channel. So your membrane depolarization is going to cause the cytoplasmic side of your membrane to become more positively charged. What's going to happen to the positive charge of your S4s? They're going to be repelled by the positive charge there, right? Okay. And so your cytoplasmic side, again, is going to become more positively charged. This is going to pull your paddles through your membrane, like you can see here. This is pulls the base of the channel apart to open the channel. So again, positive charge pulls your membranes through. And again, that change in voltage, that's going to open this channel right here. Can everybody see how your positive charge got reoriented and moved up through your channel? Well, through your membrane, along your channel. Okay. And this moving that up and away opened it up. Everybody see that okay? Normally this is like 3 angstroms, and that's not big enough for a potassium to come through, right? Okay. Anybody have any questions about that? Okay. Okay. Beautiful demonstration, right? God, that's a great figure. Okay, so the channel can be inactivated by occlusion of the pore. The ball and chain model is the name of this model. The potassium and sodium channels undergo inactivation within milliseconds of opening typically. And so this would be your wild type right here. And so we can see here that it activates, a bunch of it comes through, changes. This is your membrane current, by the way, i.e. the amount of charge moving through that membrane. Okay. And then we rapidly get to the point where it will deactivate, right? Why does it deactivate? What happens when there's no positive charge? Yeah, these are going to reorient back over, aren't they? Isn't that beautiful? Gotta appreciate that. The secrets of your life. Slowly being unwrapped. So what would happen if a mutant shaker channel lacking 42 amino acids near the amino terminus opened but did not ever deactivate? What's going to happen to that membrane current? Yeah, it's like if your switch stopped working, got fused, right? You can't turn it off ever, can you? And so it'll never turn off, and you'll just keep putting current over as fast as you can get it over. Okay? Now, eventually, actually, your cell would stop being able to put current through. You'd run out of ATP, right? To keep pumping these potassiums, and yeah, eventually this is going to stop. But in the short term, milliseconds after, you can keep going for quite a while. Okay? And that's what you see. This is the if you take that inactivation, we restore it by adding a synthetic peptide corresponding to the first 20 residues of your native channel. We can actually see that we can actually kind of get it to approximate what it looked like before, right? And it's pretty close. At least it has the same sort of activity, right? And so that's actually that first 20 residues of your native channel that's causing that open close. So this is your ball and chain model. Ball and chain model is the model for the channel inactivation, which the first 20 residues of your potassium channel form a cytoplasmic unit. This is referred to as the ball. That is attached to a flexible segment of the polypeptide, which is your chain. In the closed state, the ball is in the cytoplasm. In the open state, your binding site of your ball is actually created. And so this is what this looks like. So right here, you have your inactivated domain. And again, it has the ball right here, and that's the chain, right? Okay. And we know this has to be involved in this process somehow, right? Because what happens if we don't have those? Yeah, it never closes, right? It opens okay. So the opening part, that's functioning. This part where the ions charge causes this to switch over into the membrane, that works. This part doesn't work though, does it? Where we inactivate it. Okay? And so what ends up happening here is this ball then moves with this because the chain is attaching it, right? And so this then brings it over to the open side of this channel, right? And anybody ever played that game where it has the ball on the string and you catch it? Okay. Even if you're a monkey that doesn't know how to do it and you have the reflexes of a brick, you eventually get it, right? Okay. And that's because there's a probability that it's going to land in there no matter what, right? Because the chain itself limits the options. Okay? Same thing here. Because the chain now brought it over so that it could land in there, eventually it's going to land in there, right? And it's going to block that channel. Okay? And that's what's going to cause that inactivation, even though it's open still. Okay? And so this limits the amount that can come out by blocking it in the short term. Think of it as a bit like we're blocking it, but we haven't had time to reset it yet. Does that make sense? Okay, now the recovery of the channel. This is going to be where we're actually going to bring this charge back down in. Okay? Does everybody understand why this can't just come back down by itself, even though we've removed that charge? Like, yes, that facilitates the ability for it to come back down. If we didn't remove that positive charge, it would have to take a lot of energy to bring it back down. But just because you've taken away the repulsion on this side, that doesn't mean you've given it any energy to move, have you? Before, you gave it energy to move. Okay? Now, there is some resting energy to move that's just randomness, but that's a pretty good size group, right? And there's things to overcome when you do a conformational interchange like that. Okay, so the recovery is going to be the process of returning the inactivated channel to its initial closed state. Remember, at this point, we're in this state right here. We have an inactivated channel that is different than the closed channel, okay? This was the closed channel. Closed, open, inactivated. Okay, so this is going to require a few things. It's going to require the disengagement of the ball from the pore, opening and realignment of your paddles. This can take milliseconds to seconds. The channel cannot be opened by further membrane depolarization until you recover this. By the way, notice these numbers down here. We only ever went up to 60 milliseconds. It wasn't enough time to close this channel, right? If we would have let this go out to like a second, we for sure would have seen this come back down because we would reset our channel. Okay? Or at least we might be able to, assuming that the enzyme can actually work on it. Okay, we got this. So acetylcholine receptor. This is an archetype for a ligand-gated ion channels. This is acetylcholine right here, actually. Everybody's heard of acetylcholine, right? Okay, fantastic. Then this is going to be beautiful. What's our time look like? Okay, we'll stop here. This is actually a pretty good place to stop. Does anybody have any questions before we go, or we good? Okay, you all have a wonderful day. If anybody wants to talk to me, please catch me. We can also work on some lab stuff, and I'll send out an email Wednesday for the organic 2. You can ignore that. Following. Yeah. I mean, I could do in spring break, but I feel like that would be evil. Yeah. And an evil that's a little